Hello everyone, I'm Haley. Today I'll be presenting the paper that is titled Trace Metals and Organochlorides in the Bamboo Shark's Clethoxylon plagiosum from the Thousand Waters of Hong Kong, China. And before getting into the content of the paper, we have to know a few things about bamboo sharks. It is not like the big fast swimming sharks that you have might seen in the movie, and they hunt prey for food. Um, s- Bamboo sharks are actually the sharks are small sharks living in the shallow waters. And for their eating habits, they eat small creatures like shrimps, worms, and smaller fishes. And they are commonly caught for food, often used in soup and some fish products. This is the best picture I can find on the internet because there are many fish made with bamboo sharks that looks very terrible. And this I don't know. Oh, I cannot play this video. Um, Bamboo sharks are benthic species in Southeast Asia, and benthic means they live on the seafloor, and I actually want to show the video that they swim along the bottom of the tank, but it is it cannot play. Oh, oh, okay. And the purpose of the study, uh, since the pollution is a growing problem in coastal waters, and the bamboo sharks live in these waters. So to measure pollution accumulated in their body is the task in this study. And from the results to measure the pollutants in the bamboo sharks, we can assess the potential risk to marine ecosystems, and if people eat the bamboo sharks for food, we can assess the risk if they um through the bioaccumulation process. And the study area is the sea area around Hong Kong, including the Lamel Island. And what the researchers do in this study, they recorded the shark's lens ways to identify if it's young or old individual. And they took muscles, liver, and spleen samples for analysis. And this is the device they use to determine the elements in bamboo shark's body. It is called inductively coupled plasma atomic emission spectroscopy. And from the analysis, we can see that in the liver, silver and cadmium's concentration are the highest, and in spleen, they are copper and manganese are the highest concentration. And zinc's concentration are evenly present in all tissues. And from this table, we can see certain metals concentrate in specific organs. And it suggests that different organs have different function in processing or storing different metals. And in this table, it shows the different pollutants and in compared to bamboo sharks and other shark species. And the first two PCBs and DDTs are organochlorine compound. And compared to other kind of sharks, bamboo sharks have lower concentration in these pollutants. And there's an exception that is this PPDDT. The bamboo shark shows bioaccumulation over time, which means the older sharks have absorbed more concentration of these compounds over time. And zinc is also an exception because it should show that they have also the lower level, but it is more than the expected level. Maybe it's because of the Hong Kong seawater is being polluted by this kind of elements. And from this table, we can see bamboo sharks are not as contaminated as larger sharks species, since they live in different places and they eat different food. They might have different exposure to different environments condition. And this table is the organization of the previous two tables. And this shows most metals decrease when these sharks get older. And the pesticides increase when the sharks grow bigger. And what does this mean is that some metals might get diluted when they grow bigger. And the second one shows that these sharks cannot metabolize this compound, so it keeps absorb and store more of it. And the conclusion is that in my introduction, it says from the results we can assess the potential risk to marine ecosystems and human consumption. And from the results, we can see that eating bamboo sharks isn't very risky, since their pollutant levels are relatively low compared to other shark species. And for the environmental warnings, even though the concentration of pollutants in these sharks aren't really high, but the study shows that the pollutant is still affecting marine life. Thank you for listening.
Can you explain the different type of pollutants? I'm not sure what they are.
I forgot to put the full name on it, so
What kind of this pollutant? Like chemistry or what?
It is these three are written in the title. It is organochlorines, but I don't know the full name. I don't know the full name of each one. It is maybe it's in the paper, but I forgot to put on it.
Okay, thank you.
Can you explain the main research motivation of this PPT?
Because I am keeping this kind of sharks recently, so I think it is quite interesting doing this.
Okay, thank you.
I want to know how many bamboo sharks were they used in this study.
Oh, I don't know. But they have catch this fish for a period of 1 year. But I don't know how much sharks are there.
And how did their body size range?
The smaller one is between 20 centimeters, and the largest one can be close to 1 meter.
Oh, okay, thank you.
And can you explain again, like what's the chart means?
How it works is not matter in this study. It's just the device they use to determine the elements in the shark's body.
Okay, thank you.
Based on the food chain, I wonder that what is this species next? Bamboo shark. He is the small player, and I wonder that he accumulates about the trace metal. He can live with that.
I believe they can live with that since these sharks have been caught and been analysis. The elements have been accumulated in their body for a while before they getting caught. So I believe they can bear the elements accumulated in the body. But I don't know the limits. That's all.
So I think your talk was smooth and easy to follow, but still there are a few points for you for your improvement. The first is you were presenting. I'm sure your English is good enough to not to look at any script, but you were looking at your slides all the time. I think next time you can look at them. And the good thing of her presentation was you can see that she used the mouse very often. Therefore, we always know which point she was talking about. This is really good. And regarding the video, you were like asking why it didn't play. So if you really wanted to avoid this problem, you have to set automatically. Okay, then it will play automatically next time. At slide number 6, my feeling was your talk was not smooth at this slide. So please improve that next time. At slide 8, she used this instrument image to explain it. That's fulfill my requirement how you should introduce the instrument. So that's good. These are her results tables. She made all the tables herself. This is good because as far as I remember, the paper was full of these tables. That was not comfortable, okay, to read. So it is nice that she has condensed these complicated information into simpler table. And also at the bottom of each table, she provide a concluding remark. So we know very clearly what is the message about this table. So this is all nicely done, very nice. The main weakness comes to these slides because most of them, they don't really know what is PCBs, DDTs, PPDDT, zinc, blah, blah, blah. You probably want to say, you don't know that much either. That's fine. That's how you should introduce them. So first, because your talk was 1 minute short, so you have time to provide additional information. Like I would say, you should provide 1 slide for this, 1 slide for this together, and 1 slide for this. And for when you want to introduce it, you show their structure. You know molecular structure, right? You can find their structure at the internet. Then the most important thing is you need to tell them what is the health risk when eating too much of this. Okay, that's the key point. The same for this and this, and you probably need to tell us why this one is specifically discussed. Is it highly toxic? Is it because of that? So you talk about it. And you need to tell us what, because very often we say, oh, we need to eat zinc. So we think zinc is a good nutrient for us. Then you tell us zinc is a contaminant. Maybe you can tell us what will happen to us when we eat much zinc. That's a question I need to ask you. Increase with size. Is there any relevant figure in the paper which talk about the increase with size? It's not clear to me because this one basically has no figure except this one. Okay, I was hoping that maybe there was a figure for you to show, but it seems not to be the case. Then it's fine. 11. It seems like there's no figure in this paper. You have this good concluding remark, but I don't know if you are aware that this has different font and size. This is Calibri and this is Arial. So please, use one font. And lastly, conclusion. The conclusion here you make is mainly for implication, broader implication. But before this, please provide a slide summarizing the key finding, like what kind of pollutant is accumulated in this animal. Conclude the main finding. Then after that, provide implication. It's not dangerous to eat. That's the main implication. Okay, I think it's a good talk, but there's still space for improvement. Hope to see that next time. Thank you, Haley.